So, good evening, everyone. This is your seventh week session with me. So, in this session, we'll look into the CRISPR-Cas9 technology, which is generally the clustered, regularly interspaced short palindromic repeats Cas9 technology. So, in this, we'll go into these topics. Firstly, we'll see the CRISPR system in bacteria. Then we'll see that how we can use this CRISPR-Cas9 in genome editing. Then we'll see what are the different applications of this technology and then different computational resources which are generally used for CRISPR-Cas9 technology. Then we'll practice few MCQs and we'll also look into the current literature and then I'll take your doubts. So first of all, we'll talk about that how this CRISPR was discovered. So in 1987, there was a scientist named Ishino. So while he was studying the alkaline phosphatase genes in E. coli, he found that there are some repeated palindromic sequences which are present in E. coli. So then he checked that these repeats were quite unusual because they were separated by variable sequences of equal length. So as you can see that these pink color boxes, these are the repeat units. And then there are some spacer sequences which are 32 nucleotide in length. So he mentioned that these repeats, they are separated by equal length spacers. So which is quite unique thing, which is quite unusual to find in the E. coli. So the function of these repeats was unknown at that time. So they were treated with curiosity and their biological role was not connected to any cellular process in 1987. So he could not understand that what is the relevance of these sequences. So this initial discovery was nearly forgotten for over a decade. After that, few scientists came who looked into it deeply and then they identified that what is the role of these sequences. So this is the review which was published in 2018, which talks about all the studies done by Ishino. So if you are interested, you can go through this. Then in 1992, there was another scientist who was Francisco Mojica. He was studying the halophilic archaeon and its name was Helopherex mediterranean. So I hope you are aware of the what the archaeon means. So these are the primitive organisms which used to survive the extreme conditions. So he used it this particular archaeon, it survived in the halophilic conditions and he identified that there are 14 unusual DNA sequences which are present in this archaeon and 30 base pair long and they repeat at the regular intervals. And each sequence read roughly the same backwards and forwards. They repeated every 35 base pairs or so. So this observation is same as what Ishino mentioned, that there are some repeat sequences and they have some spacer elements which are of fixed length, which is 35 base pairs. And he also said that these sequences are roughly same backward and forward, so it means that they are palindromic in their nature. So these observations also favored what Ishino has mentioned. So he hypothesized that these sequences must have some biological function. So same as I mentioned previously that these are the repeat units and there are the spacer elements which separate these units. So he said that this is not by chance that we observe this pattern. There must be some biological function associated with it. So despite skepticism that many people at that time, they mentioned that these repeat sequences, they are present in almost every organism. So they do not have any significant role. But still, after all those skepticisms and lack of funding, he still continued his research and he kept on researching on them. So later on, in around 2002, he was working with another scientist, so who was a Dutch researcher, Ruud Janssen. So at that time, Mojica and Ruud Janssen, when they were working, so they coined the term CRISPR, which was cluster regularly interspaced short palindromic repeats. So that means that there are multiple such short palindromic repeats which are present in cluster and they have some regularly spaced, means spacer are present in between these repeats. So from there the name clustered regularly interspaced short palindromic repeats came out. And then this scientist who was Ruud Janssen, who was working with Mojica, so he also described that what the role of Cas genes is. And he mentioned that CRISPR is not sufficient to work without the Cas genes. So Cas genes also are important and they are associated with the CRISPR and then they together perform the functions. So these Cas genes, they were found consistently near the CRISPR arrays in the genomes. So he observed that these genes are always near to the CRISPR arrays in the genome. So that means there might be some association of function between them. So he said that this is the hint that the CRISPR and Cas proteins are functioning together. So their work shifted CRISPR from being the odd repeats to potentially functional systems. Because previously, scientists were thinking that these are just the odd repeats, but now, with the help of these two scientists' discovery, they said that CRISPR and Cas are performing function together. So it led to the potentially functional importance of the CRISPR technology. So in this, so these are the two scientists, and 2002 they have given the term CRISPR, and they also identified of the Cas9 genes. So in this cell, it's a prokaryotic cell. So it shows that how the CRISPR-Cas9 system works. So in stage one, for example, a virus or bacteriophage is attacking the prokaryotic cell. So how it will attack? It will insert its genetic material into the bacterial host or into this prokaryotic cell. So this is how the foreign DNA is acquired by the prokaryotic cell. Now, this component of this component of the foreign DNA, or which is coming from the bacteriophage, is getting incorporated into this CRISPR locus. So here it has got incorporated. So what is happening is that these Cas genes, they allow the foreign DNA elements to get incorporated into the host system. So in this way, the repeat element is inserted into the host genome with the help of Cas genes. Now the CRISPR locus will get transcribed because we all know that DNA converts to RNA. So that process is transcription. So when the entire host genome is getting transcribed, so at the same time, because these elements are already integrated into the host genome, so they will also get transcribed. So in this way, it will form the free crRNA, which is the CRISPR RNA. So as you can see, so these black elements, these are generally the repeat sequences, and these are the spacers which are present in between. So these are the spacer elements, and these stem loop like structures are the repeat elements. So they are, because they are palindromic in their nature, so due to which they make this secondary structure in which they make base pair with each other and form this type of structure, because as we know that RNA is generally single stranded. So after this, all of them will get separated, will get cleaved off, and there are multiple such uh, RNAs will be present in our cytoplasm of this prokaryotic cell. So now what will happen is that next time, whenever the another bacteriophage will attack it, then it will be able to recognize this particular element because it was already present in the host genome. So bacteria has now developed the machinery to recognize the self versus non-self. So now RNA guided targeting of viral element will take place because we already have these RNAs. So as soon as virus will attack it, so these are complementary to those elements. So they can easily bind and then they can target those bacteriophage uh, DNA, which will be entering into the host. So in that way, RNA guided targeting of viral elements will take place. So this was the defense mechanism of bacteria. So this was the role which was later on explored, that why these elements are present in the bacteria and how it was formed. So it's like the memory of this bacteria, memory of infection. So there was another scientist who was Volotin in 2005. So he talked about these viral DNA similarity and the PAM. So his full name was Alexander Volotin. He discovered that these CRISPR spacers, they match the fragments of viral DNA, because they were mentioning that there are palindromic repeats, and in between those repeats there are some 35 base pair spacer elements which are present. So these spacer elements, when they were identifying that what are these spacer elements, so they observed that these spacer elements resembles the viral DNA. And he suggested that CRISPR is recording the previous viral infection like a genetic memory. So whatever virus infects the bacteria, what bacteria is doing, it is taking that particular component of the foreign DNA and it is incorporating into its genome. So that particular component is now acting as a spacer. And where it is getting incorporated, it is between those repeats. So that means Cas-gen is not inserting it randomly anywhere in the genome. It is specifically incorporating these components in between the uh, repeats which they have identified. So he identified that there is a protospacer adjacent motif. Short form is PAM, which is a short conserved sequence. So there is a conserved sequence which is present here. And these PAM sequences, they are essential for Cas proteins to recognize the viral DNA. So now these PAM elements, so this PAM motif, this is responsible to identify that which one is the viral DNA. So as it is mentioned here. So this is the PAM element, and it will recognize that what is the viral DNA. And these findings strongly suggested that CRISPR plays a defensive role in bacteria. So this is the paper in which Alexander Bolotin has mentioned that how these elements, they have the spacers which are not belonging to the bacteria, but they are extra chromosomal elements, or as you can say that they are entering externally into this bacteria. And later on in the paper, they have mentioned that these extra chromosomal elements which are acting as the spacers, they have come from the viral DNA. So this shows that how the CRISPR-Cas9 defense system is working. So it is complex, which is made up of single guide RNA molecule and a Cas9 enzyme. This is the Cas9 enzyme, which is highlighted in the light blue color, and then this is the guide RNA, which is represented with this purple color. So now what happens? Phage genome, which is double-stranded DNA, enters into the bacterial cell. Now the infection is happening, and the phage genome is entering into the host cell. So a region in the phage DNA is the target of cleavage by the CRISPR-Cas9. So now there will be some region. So they have zoomed out this particular region. So now this is the region of the double-stranded phage DNA. Now what the CRISPR-Cas9 system will do? This CRISPR entire Cas9 complex along with the single guide RNA, it will come and it will look into the region in the phage DNA. It will search for it. So this is the PAM sequence which is present in the DNA which is coming from the phage. So this sequence will be recognized by this single guide RNA and Cas9, and they will come to know that yes, this is the foreign DNA which has entered, and this is not unique to bacteria. So what they will do, they will lead to the cleavage. So this Cas9 enzyme, it will cleave the double-stranded DNA which is coming from our phage. So CRISPR-Cas9 recognizes and double cuts a DNA site that is followed by the PAM sequence. So where the cut is happening, so it is close to the PAM site. And then, okay, so third step I missed. So in this case, the single guide RNA has some phage DNA sequences acquired from previous viral exposures, and these sequences are used to target the phage DNA at a certain site with the help of PAMs. So because the means bacteria already have the idea of this particular sequence which it has integrated when it was previously affected by the same phage. So due to which now it has the idea, it will have the complementary sequence to that of this double-stranded DNA. So it will be able to recognize that component and it will allow the Cas9 to make the cut. So in this way, the cut has been made, and now this fast PAM sequences are required by the CRISPR-Cas9 for recognition and selection of cleavage site. So as soon as cleavage will take place, it will affect the double-stranded DNA which has entered into the host. And so this is how it protects the bacteria from the phage attack, because later on it has already broken down. So our bacterial cell will recognize that it is a foreign DNA. So in this way, it will not allow the phage to attack the bacteria. In that way, the bacteria using the CRISPR system as a defense mechanism. So then later on, then another scientist whose name was Berengo in 2007, he looked into the adaptive immunity. So in that way, what is happening is that the bacteria are gaining the immunity. So this immunity is not inbuilt, but it is adaptive, because as the first virus attacks it and as the component of that particular virus is getting integrated into the genome and acting as a spacer, after that it is acquiring immunity. So at the first time, that immunity is not obtained or is not observed. So in that way, it is an adaptive immunity. So he confirmed that these CRISPR-Cas9, it is an adaptive immune system in the bacteria, and they also showed that the bacteria incorporate the viral DNA into the CRISPR arrays. And these sequences serve as a library of past viral infections. And when a virus reinfects, then this particular CRISPR RNAs, they guide the Cas proteins to destroy the matching DNA. And this provides the first experimental proof of CRISPR's immune function. So Berengo has done this study in 2007, it was published. So it talks about that the CRISPR provides acquired resistance against viruses in prokaryotes. So in this, they have performed an experiment, and this is just one excerpt from that particular experiment. So what they did is that this is the wild type sequence. So this shows that how the CRISPR spacers provide resistance to different phages. So in normal condition, in wild type condition, when the host genome lacks the viral DNA sequence in it, so this is the wild type genome. So now they are talking about sensitivity to FASH 858 and FASH 2972. So these are two different strains of two different phages. And when we say sensitivity, that means how sensitive is my bacteria to the attack of these viruses. So if sensitivity is high, that means my virus is very sensitive and it will die if some virus will attack it. Or the virus can get the capture of the host system. So sensitivity is high, that means the bacteria is more prone to get infected. So when it is wild type, so wild type is the case when there are no spacer elements which are inserted. So S1, S2 represents the spacer elements which are coming from the viruses. So when there is no spacer, then the sensitivity is maximum in both of them. So which means no new spacer sensitive to both the phages. Now if we look into the S1 and S2, so here there is gain of the spacer elements. So you can see in this they have talked about that the wild type 5858 plus S1, S2. So when we say wild type along with the 5858, that we mean that when the 858 patch has infected it and from there it has incorporated this spacer 1 and 2 into its genome. So now what will happen because the spacer belongs to 5858? So now it will gain sensitivity against this virus because it already have the knowledge of the genetic element of this particular virus. So as soon as this virus will infect the bacteria, because bacteria already have the identical sequence to that of virus, so it will identify that this is the viral DNA, so it will attack it. CRISPR-Cas9 complex will attack it and protect the bacteria. So in this way, sensitivity has decreased. That means bacteria can fight against that virus and it's a bit safe. Whereas if we talk about 52972, so because there is no some spacer information of this particular virus, so still the bacteria is sensitive to this particular bacteriophage. Now we'll see in this case. So in this case, the wild type sequence is there and the spacer is obtained from 52972. That means now the 5272 has attacked it and it has the spacer obtained from this particular virus. So now you can see that the sensitivity to this particular virus has now decreased. So why it has decreased? Because it already has a sequence which is identical to this viral DNA, so it can attack it. So in this way, in this experiment, they have shown that how the incorporation of spacer elements can allow the bacteria to fight against these viruses and its sensitivity decreases. So it can survive to those infections. That means this is leading to the adaptive immunity in the bacteria. But as you can see here, that this is the adaptive immunity. So in the first time when these viruses will attack, then the bacteria will be sensitive. And with sensitivity, I mean the viral DNA will get incorporated into the host and then it will try to take up the machinery of bacteria. But at the same time, it might not kill the bacteria, but it might compromise the what how the bacteria was surviving previously. So in that way, this is how the adaptive immunity is gained through the attack of the viruses. Now, now we'll talk about the mechanism of bacterial immunity. So same thing we'll talk in detail now. So these CRISPR arrays, they consist of repeats and the viral derived spacers. Now, because we know that those spacer elements, they are derived from the viruses. So when a bacterium survives the infection, this viral DNA fragments are captured by Cas1 and Cas2. So what will happen is that because virus is attacking the bacteria, so in many cases bacteria might die because that virus will compromise the needs of bacteria. Bacteria might not get enough nutrition for itself, so it might die. But the bacteria which survive this particular infection, because now the viral DNA will be integrated into the bacterial DNA because that is the nature. That is the nature how the viruses attack bacteria. So now they have those viral DNA in them. So these will be captured by the two proteins. So as you can see that viral DNA is processed by Cas1 and Cas2 and that is incorporated into the bacterial DNA. So this is how the spacer is created in between the repeat sequences. Now in the next step, these fragments are inserted into the CRISPR arrays as new spacers. And the CRISPR RNA are transcribed from these arrays during the reinfection. So now this particular bacterial DNA, when it will get transcribed, so for all these spacer elements, the RNA will be synthesized and this RNA is known as CrRNA. So till the time it is not separated, it is referred to as pre-cRNA. That means this is precursor CRISPR RNAs. So how it will express? So afterwards, whenever the reinfection will happen, then these precursors crRNAs, they will get processed by the RNase 2, RNase 3. So RNase 3 is an enzyme which will break down, but means which will separate these RNAs which belong to different spacers. So in that way, we will get separate crRNAs which are specific to specific viruses. So in this way, it is processed. Now the next step will be ribonucleoprotein complex. So this crRNA guide the Cas proteins to target the viral DNA for cleavage and destruction. So now from all of them, now they will act as the guide RNA. And how they will act as a guide? Because for example, the same virus attacks. And let's assume it belongs to this red colored spacer. So this particular virus. So now this will act as a guide RNA. It will go to Cas9 and it is as it is the component obtained from the same virus. So its sequence will be complementary to this virus. So it will make a ribonucleoprotein complex and it will be hybridized to this particular genetic element. So hybridization will take place, and with the help of Cas9, there will be a double strand break in this double stranded DNA of the virus. So this is how hybridization and cleavage takes place. So in this way, this breakdown or this double strand break is only possible because of this guide RNA. Because of this complementarity, that is how the Cas9 is able to recognize this site. Otherwise, without the guide RNA, Cas9 is insufficient to identify these sites and make the double strand breaks. Yes, these are the two phases that how the mechanism of bacterial immunity works. First stage is the immunization when the spacers are made, and second step is the defense. When that same virus attacks, then these spacers along with Cas9 identify that sequence and breaks it. So we'll talk about little bit about the history of CRISPR, that how it began. So in 1987, the discovery of CRISPR repeats by Ishino in E. coli, but at that time he could not identify that what is the significance of these repeats. So in 2002, the term CRISPR was coined by Mojica along with Jensen, and they have also identified the Cas genes, and they mentioned that these two function together. Then in 2005, the spacer sequences, so Bolletin was working and he said that these spacer sequences are extra chromosomal and they are not belonging to bacteria, but they are coming from some external sources like viruses. So in that way, when he was checking that to which sequences these spacer elements belong, so he identified that they are highly homologous with the viruses. So that means might be when virus infects, so these sequences get trapped in between the CRISPR repeats in bacteria. So in 2007, when people were looking at the what is the significance of these CRISPR elements, then they identified that it has the role in adaptive immunity. So Berengo has confirmed it. So in the experiment, as we have seen that how when spacer was not present, then our bacteria get infected by the viruses. But as soon as the virus attacks it and bacteria survives, then bacteria already have the information of that genomic element of that virus. So next time when the virus attacks, then this particular bacteria that it already has the genetic element, so that genetic element gets transcribed into the CRISPR RNA, and it along with Cas9, it attacks the incoming viral DNA now. So in that way, it gains the immunity against that particular infection. So in 2012, CRISPR-Cas9 was engineered for the genome editing in vitro. So then this particular technique was also explored in vitro, that how we can edit the genomes with the help of this CRISPR-Cas9 technology. In 2013, eukaryotic cells were modified by using the CRISPR-Cas9. So now what we can understand from here that Cas1 and Cas2 genes, what they were doing, that as the viral element was entering, it was making sure that this element gets specifically incorporated in between the repeat elements. So what we can do, that we can also explore this system CRISPR-Cas9 and insert anything in between the host genome. So in that way we can edit the genomes. So that's why in 2013 the eukaryotic cells were modified using this technique. In 2014, discovery of the precise methods to modify gene in eukaryotes with CRISPR-Cas9 was made. In 2016, oncogene mutations were inactivated by CRISPR-Cas9. So now if you know that oncogenes are the genes which are responsible for the cancer, so if there is some mutation which is making the gene oncogenic. Because normally the genes are the same in the DNA. There are no new genes are coming into the system. But what is happening is that if some gene has some role, which is a balanced role, but if there is some mutation in that gene, then that gene behaves differently. It might get overexpressed, underexpressed, or it might lead to loss of function or gain of function. So in that way, these mutations can lead to cancer. So if there is some mutation, so we can inactivate that mutated gene with the CRISPR-Cas9. In 2016, it was observed. Then in 2017, CRISPR-Cas9 was used to modify the beta globin gene in the human embryos. So in that way, we can treat it. So in future, if we know that this particular child might suffer from this disease, so in the embryo stage only, we can cure those genes. So this means we can somehow doing the gene therapy. In 2019, He Jiankui was jailed for deliberately violating the medical regulations and for rashly applying gene editing technology to human-assisted reproductive medicine. So now there are some ethical grounds which exist. We cannot use this on any organism like this only. We need to follow some rules and regulations. We need proper permissions. We need proper channels. So He Jiangi was jailed because he rashly applied those gene editing technologies without following the rules and regulations. So this thing we always need to keep in mind that these techniques, although they exist, but we still need to follow some rules. We cannot directly go and edit everything, or we cannot directly test it on the human samples or human organisms directly. So there are some rules. In 2020, Emmanuel Charpentier and Jennifer A. Doudna were awarded the Nobel Prize for the development of method for genome editing. So they have mentioned that how the CRISPR-Cas9 system can be used for the genome editing. So they were awarded the Nobel Prize in 2020. Yeah, and if you're interested to read more, then this is the review in which they have discussed one by one different technological advancements which were made, how it was identified, how its structure and function was characterized, and now what are the applications. So till 2020, everything is mentioned in this review. So if you're interested, you can go through this. And these everything whatever has mentioned. So we have discussed, we have covered all of them in the previous slide only. Now we will talk about the classes and types of CRISPR, that how many classes of CRISPR exist. So this CRISPR system can be divided into two classes. So when we say class 1, then it means it is a multi-protein complexes, such as type 1, type 3, type 4, so in which multiple proteins are involved. Because previously we were saying that CRISPR-Cas9, that means only Cas9 is involved. So that is a single protein effector. So class 2 comes under single protein effectors such as type 2, type 4, type 5 and type 6. Type 1, type 2, type 5, they target the DNA. So type 1, type 2, type 5, they target DNA. Type 6, which is from class 2, target the RNA. And type 3 from class 1, it targets both the DNA and RNA. So when we say target, then we mean that because the viral genome, it can be RNA virus or it can be DNA virus. So in that way, the CRISPR systems are class 1 and class 2. So depending on what kind of virus is attacking, so that particular type of CRISPR system will attack it. So if it is a DNA virus, then type 1, type 2, and type 4 will be the CRISPR complexes which will attack it. And if it is the RNA virus, then type 6 will target it. So different classes use distinct Cas proteins. So mainly we talk about Cas9, but there are other Cas proteins also which exist, such as Cas3, Cas9, Cas12, and Cas13. So when we say class 1 CRISPR system, and these are the types, type 1, type 3, and type 4. So as I mentioned that these are the multi-protein complexes. So typical protein is Cas3 in case of type 1, Cas10 in case of type 3, and CSF1 in case of type 4. But along with these proteins, there are other proteins also which are helping it, such as Cas6. In this case, Cas6 and some unknown factors, and in type 4, it is unknown. This protein is not yet identified, but it exists. So that means multiple proteins are involved in this class 1 CRISPR systems, and they target the DNA. Type 3 can target both DNA and RNA. Type 4, it's not well discovered. Now self versus non-self discrimination. So how it discriminates. So type 1, they discriminate with the help of PAM, which we discussed. which is the spacer element. So here, when we were talking about the PAM sequence, this is the protospacer adjacent motif. So this is the PAM. So not all the class of CRISPR identifies the foreign DNA with the help of PAM. So in this case, it is the PAM with which it distinguishes between self versus non-self. Whereas in case of type 3, it uses the CRISPR repeats. So with the help of those repeat sequences, it will be able to identify that which is the self and which is the non-self. Because if it is a self element, then it will not be surrounded by the CRISPR repeats. So if any element is surrounded by CRISPR repeats, then it means that particular central element is my non-self element, or you can say the viral tracer element which is present. These are the effectors. So effectors are the other proteins which are helping it out. So these are series of such proteins which are involved. Now in class 2, when we say type 2, so in this case, Cas9 protein is involved, in type 5, CPF1, and in type 6, C2C2. And this type 2, class 2 of CRISPR systems, they mostly target the DNA, and in this case, PAM are the regions which help the bacteria or which help this CRISPR-Cas9 system to distinguish between the self and the non-self. And then these are the effectors. So Cas9, CRISPR RNA, and tracer RNA. So these all are required to perform this function. So as I mentioned that in this case only single enzyme is enough along with the CRISPR system. And similarly in type 5, CPF1, and it also targets the DNA and it recognizes the self versus non-self using the PAM protospacer adjacent motif. And in case of type 6, which is not well studied, so in this case the protein is C2C2. What is the precursor chromosomal RNA processing? So that is not yet known, and what it targets is also not yet known. So this is how we can distinguish between both the class of CRISPR systems, and mainly for genome editing, we will mainly focus on this class 2 type 2 CRISPR system. So you will see that we will be talking about CRISPR-Cas9, and the RNase 2 is used for the processing of this precursor cRNA, where we have seen that how different RNAs which contain different spacer elements are separated into separate guide RNAs. So for that particular cleavage, RNase 3 is required. It targets the DNA and then it recognizes PAM motif, after which the Cas9 will cleave that particular double-stranded DNA of virus. Okay, now we'll talk about the Cas9 and the tracer RNA and type 2 systems, because I mentioned that we will be mainly focusing on the type 2 systems where only single enzyme is used. So type 2 CRISPR systems, they are defined by Cas9. So type 2 and class 2. So I will be talking about type 2, which is belonging to class 2. So type 2 CRISPR systems, they are defined by Cas9, and these are large 1600 amino acids endonucleases. So when we say Cas9 and we are saying that it is bringing about double strand break, that means these are the enzymes, these are the nucleases. So Cas9 is a large nuclease. So I just want to give you a brief recap. Then when we were talking about the telins, or when we were talking about the zinc finger motifs or zinc finger nucleases, then we mentioned that along with those elements which are DNA binding, we are adding nucleases. Similarly, in the case of zinc fingers also, and in the case of tail also, which was the transcription activator like effector molecules. So with tail also we add nucleases, it becomes talin. So in both of them, we were adding nucleases so that we can bring about the double strand breaks. So similarly, in case of CRISPR system also, we also need the nucleases. So in this case, the nucleases which we use, so in the previous systems, we used FOC1. Here we are using Cas9. So these are endonucleases and these are large. And these Cas9, it requires tracer RNA to process the CRISPR RNA precursors into the functional guides. Okay, so together tracer RNA and CRISPR RNA guide the Cas9 to specific DNA sequences. And Cas9 then introduces double strand breaks at the target site, and this mechanism is the foundation of CRISPR-Cas9 gene editing. So what it is saying, that we also require this tracer RNA to process the CRISPR RNA precursors into the functional guides. So this is the CRISPR RNA precursor. So as I mentioned that in the precursor CRISPR RNA, we have different genetic elements belonging to multiple spacers and we want to process it. So in the type 2 system, generally RNase 3 is the one which breaks it down into different CRISPR RNAs, which will be further acting as a guide RNA. So it is saying that it is not enough to have just the CRISPR RNA. So this, for this RNase 3 along with the tracer RNA is also working here, and tracer RNA is also coming along with RNase 3, and it is leading to the conversion of these precursor CRISPR RNA into the proper guide RNAs. So as you can see, this is the CRISPR RNA and this is the tracer RNA which is binding to the CRISPR RNA along with Cas9. They all will make a complex, and this entire complex will help to make the double strand break in the viral DNA. So these black lines, which are the double stranded DNA of the virus, so it came here. Now what Cas9 will do? Cas9 will move along with its CRISPR RNA and the tracer RNA, and because in the tracer RNA we have the spacer, which is specific to the, which is complementary to the viral DNA, so it will make base pair with this. Now Cas9 has identified that which one is the viral genome, so now viral genome is identified. Now Cas9 will make the cleavage, so this NGG represents the PAM, which is the photospacer adjacent motif, so it will make cut near to this PAM site. So here is how the cut is made. So this shows you, so in this I have explained that what it shows. So first, in this first figure, it shows that the tracer RNA plus CRISPR RNA precursors are processed by RNase 3 and Cas9 into the mature CRISPR RNA and tracer RNA complexes. So as you can see, it is a complex between CRISPR RNA, tracer RNA and Cas9. Now Cas9 uses this RNA duplex because you know that RNA is single stranded, but tracer RNA is base pairing with CRISPR RNA, so it has converted to a duplex. So this RNA duplex recognizes the DNA target. DNA target means the DNA sequence of virus which is attacking the bacteria next to the PAM NGG sequence. So this is the photospacer adjacent motif which is recognized by Cas9 to distinguish between the self versus non-self. Now Cas9 will cleave the DNA using its RUFC and HNH nuclease domains. So this Cas9 protein has these two domains, RUFC and HNH, so generating a double strand break. So this is how the Cas9 and tracer RNA are working along with the CRISPR RNA and making the cut in the viral genome. Now this CRISPR-Cas9, it can also help in the genome editing, so it acts as a genome editing tool. And if we talk about today's time, then mainly if we compare among ZFN, TELENs and CRISPR, then CRISPR is most commonly used. It is widely used because it allows the precise modifications in the cells and model organisms. It is cheap and it is simple. Then the zinc finger nucleases and TELENs, it is rapidly adaptable to new targets using simple RNA designs, and its application includes medicine, agriculture, biotechnology, and research. So this is how it looks. This is the guide RNA. This is the Cas9. And in this, guide RNA is recognizing, this is the spacer element of that particular guide RNA, which is complementary to the DNA of virus which has attacked. Now it will make base pair with this. And after that, we'll look for the PAM site. So for example, if this is the PAM site, then Cas9 will cleave few base pair downstream of the this PAM sequence. So in that way, double strand break will take place. Now this double strand break can either lead to disruption of the gene or we can insert a gene. So in this case, we are talking about the systems where we can edit the genome. So we are not talking about the virus and bacterial systems. We are saying that how we can manipulate some DNA with the CRISPR-Cas9 system. So in that way, for example, I want to edit the kinase genes. So what I will do, I will construct a guide RNA which has some sequences which are complementary to my kinase gene so that I can make the mutations or insertions in that particular gene. So as soon as I will enter this RNA, it will be having the complementary sequence to that particular DNA. So it will bind to it and it will make the double strand break. Now I can use the enzymes and I can insert the gene of interest. So I can give the homologous sequence so that the homologous base recombination takes place. So in this, I will not be explaining it in detail because we have already seen in the previous sessions that how the repair mechanisms work. So one mechanism is non-homologous end joining and another mechanism is the homology based repair, in which we need the another sequence which is homologous to our given sequence so that the repair can take place. So similarly, with the help of the homologous end joining, it will insert the newer part of the gene or the entire new gene into the sequence which we have cut, so where we have made the cut. So this is how CRISPR can be used for editing the genome. Now we will talk about the guide RNAs. So natural CRISPR, it uses the crRNA, which is the CRISPR RNA, and tracer RNA to guide the Cas proteins. So this is the natural system. So researchers have engineered the single guide RNA combining both these functions. So when we say CRISPR-Cas9, so in that case, whenever we draw it, we see that there is only a single guide RNA which is present. So previously these two were making a duplex, but now because researchers have identified that we don't need two separate RNAs, but we can combine both of their features in just one RNA. So they have made a single guide RNA. So in single guide RNA, we will be having a sequence which is corresponding to the tracer RNA and another sequence which will be belonging to the CRISPR RNA. So in this CRISPR RNA, this sequence is like the spacer sequence, which is complementary to the gene which I want to attack on, or which I want to cleave, or I want to edit. So in that way, both of them are linked to each other and they are combinedly making a single guide RNA. So I don't need two separate RNA molecules. Then single guide RNAs simplify the system for lab use because it is simplifying it. I don't need two separate RNAs. And chimeric single guide RNAs can also include repair templates. RNA sequences determines that which DNA site is targeted. So why I need these single guide RNAs? Because they will, this target specific CRISPR RNA is the one sequence which will decide that where I want my Cas9 to go and bind in the genomic DNA. And this shows that how the guide RNA has. So this has 5 self-cleaving ribozyme site and this is also the 3' self-cleaving ribozyme site. So in this way, cleavage will take place, and after the self-cleavage, we will obtain the guide RNA. So this will be my guide RNA, and both these sites are now cleaved off. Yeah, and this also shows the same thing, that this is the guide RNA, this is the Cas9 complex, and in the guide RNA, we have a target-specific CRISPR RNA sequence, which is complementary to our genomic DNA. And this is our PAM, which is the protospacer adjacent motif, with the help of which we can distinguish between the self versus known self. So this is how the guide RNAs were synthesized versus what they naturally exist. So now we will talk about gene knockouts, that if we want to do the knockout using the CRISPR system, then what we can do. So Cas9 will cut the DNA to create a double strand break. So this is how double strand break will take place and it will be close to the PAM site. The cells will repair it using the non-homologous end joining. So this is how non-homologous end joining will take place. Non-homologous end joining is error-prone and it will lead to insertions or deletions. So this is the modified double-stranded ODN. With the help of electroporation, we can enter this externally genetic element into the cell. Now this is the repaired genomic DNA. So as we mentioned that in non-homologous end joining, it can either lead to deletion of some elements or random insertions. So in this case, there is an insertion which has taken place. So accuracy evaluation by high throughput sequencing. So in this case, this is the accurate repair when the no indels have taken place. This is indels in the double-stranded ODN. Okay, so this box represents the insertions and this dark box, which shows the deletions. Now what they have done is that they have added this external element into the genomic DNA, which is being repaired. After the cut, they have integrated it inside the genomic DNA. Now they are checking that whether the deletion has been taken place in between it or whether some insertion has taken place. So in this, they are saying that there are indels in the double strand ODN. So this is my sequence, and there are some insertions which have taken place, but my genomic DNA is intact. There is nothing happening in the genomic DNA. In this case, they observed that there are indels, but in the genomic DNA. So which could either be insertions or deletions. And similarly, in this case, they observed that both in the ODN, double-stranded ODN, as well as genomic DNA, there are insertional deletions which are present in both of them. So indels can disrupt protein coding and which can lead to the gene knockout. And it is useful for studying the gene functions in disease models. So this is how we can study the function of genes, because CRISPR-Cas9 can lead to the knockdown of the genes. Now we will look into the gene knock-ins, in which we use the homology-dependent repair, the homology-directed repair. So instead of random repair, homology-directed repair can insert the new DNA, and a donor DNA template with homology arms is provided. So for homologous directed repair, we need something which is homologous sequence. So we need to provide that homologous sequence, and the homology arms are also important, because we know that if we want something to get inserted, then it should be flanked by the homology arm on both the sides. And these homology arms will have the sequence similarity to that of my genomic DNA. So both of the genomic DNA, they will share the fragment between the homology arms. It will get replaced. So this is how the homology dependent repair works. So the cell copies this template into the break site. So wherever the break is present, we have a repair template, and we'll see where the homology arm is present. So anything in between the homology arm will get exchanged with this. So here they are showing that there is a sequence change. So this sequence has got integrated over here with the help of homology-directed repair, and it allows the precise insertion of new genes or mutations. So we can also insert certain mutations into it, and it is useful in creating the disease models or therapeutic corrections. So for example, if I know that in case of sickle cell anemia, there is just change in the one amino acid, which leads to that particular phenotype. So what we can do, we know that where the mutation has taken place. So we can target that gene, and now with the help of homology directed repair, I will provide a repair template in which that particular mutation is now corrected. So after the repair takes place, my gene will get corrected in the end. So similarly, in this case, they are showing the standard editing. Here they are showing that the PAM is blocking the mutation, and in this case, the guide is blocking the mutation. So this is my CRISPR-Cas system. They will cut here near to PAM site. This is the double strand break which will be observed. Now repair will take place, but for homology directed repair, I need a repair template, and there should be some sequence which will get exchanged with the double stranded break region. Now the sequence has been incorporated along with the sequence change which I mentioned. We will re-edit it if we want. So for re-editing, we can again use the Cas9 and guide RNA complex. It will again made a cut close to the PAM site. Now we can use NHEJ to inaccurate knock-in or gene corrections. We can add, it can lead to the indels, which are insertional deletions. So this is the re-editing. Similarly, here also it has been shown that this is PAM, and here the cut has been done. Now the repair template is coming. So this is the PAM blocking mutation, because it is lying at the same complementary region to that of the PAM. So after homology directed repair, this particular mutation will exchange the PAM sequence. So previously PAM sequence was retained, but now because this particular mutation is happening at the PAM site, so now PAM site is replaced with this particular region of the repair template. Now re-edit is blocked. So we cannot re-edit it, because we know that for re-editing we need the guide RNA and Cas9 system. But to distinguish for Cas9 or for the guide RNA to make the cut, it requires the PAM sequence. That is the main criteria with which it identifies that okay, we can now cut few base pairs downstream of PAM. But as here we have made a mutation in which the PAM is replaced, so Cas9 cannot recognize this PAM motif, so it cannot cleave it. So now re-editing is blocked. So re-editing can't take place, which will take place here. So in this case, there will be accurate knock-in and gene correction. Now in this case, guide blocking the mutation. So now there is a repair template. So the topmost processes are the same, which is taking place here. Now double strand break has taken place. This is the PAM site, and this is the repair template. So this is the guide blocking notation. Now what happens is that PAM is required for Cas9 to identify it and make sure that where to cut. But at the same time, we also require the guide RNA, which is recognizing the site in the double-stranded DNA, which is complementary. That's why only the guide RNA will bind and then Cas9 will cut. But now if we make a mutation in the region where the guide was previously binding, so now my guide will not be able to bind. So here also re-editing is blocked. So in this way, there is accurate knocking or gene correction. That means my gene is accurately um, incorporated into the DNA. But in this case, if my PAM is still available, then there are chances that Cas9 can again come and make the cleavage. So in that cases, there will be inaccurate knocking. So to avoid such conditions, we make sure that when we are editing our genome, then we make sure that either the PAM is blocked or the guide sequence is blocked by mutating them. Now we will talk about the germline editing. So CRISPR edits introduced in the embryos or gametes, because we mentioned that during the gene therapy we can all even treat at the level of embryos, so that the child who develops, he should not have those inherited mutations or those disease genes in him. So we can treat it at the level of embryos or gametes. It produces offsprings with permanent and heritable modifications. So these modifications will be permanent and then heritable. So for example, if somebody's grandfather was suffering with sickle cell anemia, and now the father, so when he was born, then he has undergone this CRISPR editing thing. So what has happened to that father, that now there are the mutations in the gene, so mutations are repaired at the level of father. So now what happens whenever his son will take birth, so in that case these mutations are also heritable, so his son will also not have sickle cell anemia. So that means you have treated it and you have made it heritable. So none of his offsprings will develop the symptoms of sickle cell anemia or will have that faulty gene. So that's why CRISPR-Cas9 editing technology is so helpful. It is used in animals to create the knock-in and knock-out disease models. So we can also study by knock in some genes or knock out some genes to study their roles, that how they are influencing the disease state, whether they are leading to disease or whether they are helping in the disease state. So demonstrated in mammals for research purposes, and it raises the ethical debates when applied to human embryos. So now, because there are some ethical concerns, people do have their beliefs that something which is present in the human, they are for his best. And if we keep on editing it, then it might interfere with the balance which is present in the nature. So I'm not talking about diseases here. Many people, if they know that we can do the editing of the genes, so what they are doing is they are editing them in such a way that the person can look better or the person have better thinking capabilities. So there are many genes which are related to growth hormones or for the intelligence or many other factors for the beauty. So if we keep on editing them, then we will disturb the naturally present genes. So in that way, there are ethical debates which exist that whether we should apply it on human embryos or not. Okay, so this shows that there are therapeutical applications also and biomedical applications also of this CRISPR-Cas9 technology. So in therapeutic, the applications are applications in oncology, which means in cancers, because there are certain genes which are oncogenes which leads to cancer. So we can treat those genes. We can edit those genes by genome editing and editing the epigenome. So epigenome means when the surrounding components which are present in the environment, when they affect the expression of the gene. So in that way, I can take an example that we have different tissues. So some genes are expressed in one particular tissue. There are some genes which are only expressed in kidney, whereas other genes are getting expressed in the liver. So which means there is an epigenome. There is an external environment which is controlling the expression of the genome, which is above this genome. Because our DNA is same in all the tissues. It is not that in each tissue we have different DNA and different genes. We have same DNA throughout all the body. So in each tissue, there are some epigenetic factors which are present, which are controlling which genes to express and which not to express. So if we edit that epigenome, then we can decide the expression of genes. So this can be done with the help of CRISPR technology. Then combating infectious diseases. So this can also be uh, performed with the help of CRISPR-Cas9 technology. Now, if we talk about biomedical application, then first is regenerative medicine and tissue engineering. So this can be done. Disease modeling, disease can be understood. Medical diagnostics. So medically also we can use it as a diagnostic measure. We can, for example, if I have a sequence of some virus, and if I use that CRISPR thing and enter it into the host, so it will only bind if that complementary sequence is present. So if the it is binding, that means I can identify that yes, this virus has attacked the person. So in that way, we can do diagnostics. Then cell reprogramming, we can reprogram the cells, and drug target discovery are the other applications of CRISPR-Cas9 technology. And in this case, these are the different cells which are mentioned at the germ line. So how we can obtain the testis biopsy, then we can obtain the spermatogonia, SC stem cells derivations, and then CRISPR-Cas9 editing in spermatogonia stem cells gamete differentiation, corrected sperms, and then ultimately we will be able to correct the genes at the level of germline. Similarly, if the sperm is affected, then we can also perform in vitro fertilization and ISCI, and then CRISPR editing can be used in the embryos. So in the end, we will get the corrected embryos. So this shows that how we can use the CRISPR-Cas9 editing to treat the genes at the level of germline. Similarly, we can use skin biopsy. We can use the conventional stimulation affected oocytes or GV oocytes. So these are different cells which are collected, and then this shows that at what step we can use the CRISPR-Cas9 editing to manipulate these cells and to treat them. Now comes conditional knockouts. So conditional knockouts means that you are knocking out the expression of the gene conditionally. That whenever some reagents are present or at some particular time point or in some specific tissue, so there are conditions that when to do the knockout. So it comes under conditional knockouts. So not all knockouts are viable. That means there are some knockout. If I knock out some gene which is important for survival, so my organism will die. So which means that not all the knockouts are viable. So I cannot do, because there are some genes which are supposed to express in certain tissues. And if I knock it down in all the tissues, then my organism can die. So in that way, what I can do is I can do conditional knockout, in which I can knock out that gene in specific tissue, but letting it express in all other tissues so that my organism can survive. So it uses the LOXP sites and CRISPR to enable the specific gene deletions. So we have already talked about the LOXP sites, that how the Cre and LOX3 recombinase identify the LOXP complex and how it leads to insertion, deletion, or transversion of the genetic elements. So similar thing we'll talk here. So LOXP sites and CRISPR together brings about the conditional knockouts. Cre recombinase can activate the knockouts in certain tissues. It allows the temporal or spatial control of gene inactivation. So temporal means which is time dependent, spatial means which is tissue dependent, at which space it is taking place. So it is widely used for study the essential genes in vivo in original systems. Since we are not performing the experiments in lab, we are performing it in the host. For example, we are performing it in the rat. So this is the example. So step 1 to 40. So in this, they are preparing the CRISPR components and long single-stranded DNAs from day 1 to 15. So now we have a CRISPR RNA sequence and a tracer RNA. So as we discussed that in nature, CRISPR RNA and tracer RNA both exist and they make a duplex with each other. Although in lab, scientists have modified them and made a single RNA element, which is known as single guide RNA. But in nature, both the CRISPR RNA and tracer RNA exist. They, along with Cas9, they make a complex, which they have now referred to as CRISPR tracer RNP, which means both the CRISPR and, sorry, I need to rejoin. Okay, so now here, now they have made a complex. So this CTRNP means CRISPR RNA, tracer RNA. RNP means RNA protein complex. So ribonucleoprotein, which is made up of RNA along with protein. So this is my complex, which is combining CRISPR RNA, tracer RNA, and Cas9 protein. Now we need a single-stranded DNA, which is the repair donor. So it's left arm, it's right arm, and then this is a new sequence. So left arm and right arm, we means homology arm, which is responsible for the repair mechanism, because this Cas9 and CRISPR tracer complex, it will make double strand break in the DNA, and that particular DNA site which is complementary to my CRISPR RNA sequence. So now this is my target exon which I want to target, and these are the LOXP sites. Or I can take protein expression and fusion cassettes, or we can take artificial microRNA cassette. So these are three different cassettes which are present. Now from steps 41 to 55, so this is known as transgenesis experiments, starting from day 16 to 36. So in this case, what they have done is they did the micro injection of the CTRNP complex along with the single-stranded DNA, which is the repair donor. So this is my repair donor, which has been incorporated. With the micro injection, we can directly enter these components into the cell. So embryo transfer has taken place into the rat because we are studying it in vivo. So now from steps 56 to 60, this is the genotyping of offspring. So we will perform the genotyping. Now these are the founder mice. So this is the wild type, and these are the targeted mice. So we will compare the expression in both of them. So in the targeted mice, the new sequence is present, which is obtained from here. And in the target site, these are the sequences which were previously present. So the mice which are targeted, they all have now the new sequence. So PCR is performed to amplify the sequence and to know that whether the targeted mice now have the new sequence or they still have the target exon sequence only. So these are the experiments which shows that how the conditional knockouts are performed. And these are the steps in which we did not strictly talk about conditional, but in this case, we have talked about that how it works, how the LOXP sites and the new sequences helping to exchange the fragment in between. And with the LOXP sites, it is made conditional because we will only enter the Cre recombinases into those tissues. We'll do the micro injection only to those tissues where we want the expression to take place. Otherwise, we'll not enter it into those other tissues. So that is the main concept behind this LOXP sites. Now we will talk about the CRISPR in gene therapy. So it has the potential to treat the genetic disease at the root cause. And clinical trials are also underway for the Duchenne muscular dystrophy, sickle cell anemia, and thalassemia. So these are three major disease for which the clinical trials are undergoing by using the CRISPR technique. So their goal is to repair the faulty genes or silence the harmful mutations. So delivery remains a challenge, viral versus non-viral methods, because we have discussed that to incorporate them, there are different ways. One is that either you can use the physical methods like electroporation, microinjection, or you can use the viral agents versus non-viral agents with which you will deliver these CRISPR-Cas9 system into the host. So that is still a challenge, and it represents a paradigm shift in medicine. So first of all, it shows you different delivery methods that you can use adeno-associated viruses, adenoviruses, lentiviruses. So what you can do, you can put this system into these viruses, and then as soon as these viruses will attack the cells, and they will, along with their DNA, they will also integrate this particular CRISPR-Cas9 system into the host. Then in non-viral vectors, we have DNA nanostructures, we have liposomes and polymers, nanomers. We have cell-penetrating peptides, and then in physical methods, we have electroporation, microinjection, iTop, and mechanical cell deformations. So now this shows you that how the Cas9 complex is present. This is my target DNA, and then PAM sequence is present in the target DNA. This is the single guide RNA, and in single guide RNA, we have the component, which is the CRISPR RNA in this, which is complementary to my genomic DNA, or you can say target DNA. Now matching genomic sequence. So what the Cas9 will do, Cas9 will cleave to sequence downstream of the PAM sequence. So there will be double strand break, so which is known as Cas9 mediated double strand break. Now it can enter into either non-homologous end joining state or homology directed repair. If it will enter into non-homologous end joining, then it will disrupt the gene. It can lead to nucleotide deletion or nucleotide addition. So in both cases, the DNA will get disrupted, the gene will get disturbed. Whereas in case of homology-directed repair, it can correct the gene of interest. So in this case, what we need is we just need a homologous sequence with which my double strand break containing DNA can get repaired. So it should have the homology arm. So in both of them, whatever is present between the homology arm will be exchanged between these two strands. So now my target DNA will acquire the segment which is present in between the homology arm of my donor DNA. So in this way, we can repair the DNA. So this is how the CRISPR technology is working in the gene therapy. And this is a paper in which they have mentioned that how the CRISPR-Cas9 gene editing is like a new hope for the Alzheimer's disease therapeutics. So similarly, in case of Alzheimer's disease also, there are accumulation of the beta plaques. So in which different amyloid beta, they make a cluster. They combine together, so they make like a plaque in the brain. So as you can see, distinguish here that this is the Alzheimer's disease brain, as these are small plaques, dark gray colored. So early onset, when it has just started, then there is this is known as familial AD, that is Alzheimer's disease, and these are the genes which are associated. And then there is ApoE4 gene, which is also responsible. And then in this case, it is a known familial Alzheimer's disease. Then we can identify it using the GWAS identified loci. GWAS means it is genome-wide association studies in which we compare different genomes of different organisms, not different organisms, but different humans. We will compare the normal genome with the patient genome, and then we'll identify that where the mutations are taking place, where the differences are. So with the help of GWAS studies, we'll be able to know that which sequences are changing, that means which genes are changing. So in that way, we can identify the genes which are involved in the Alzheimer's disease. And with the help of CRISPR technique, we can specifically target those genes. And we can either correct the mutations, or we can either change those genes. If that gene is lost, then we can add the new gene, or we can add the correct copy of that gene, so which can lead to gain of function. Or if some gene is getting overexpressed, then we can also make a mutation so that it get repressed. So in that way, it will lead to loss of function. So these are the byproducts which are obtained through the genome editing. And in this, they have mentioned that how, with the help of CRISPR-Cas9 editing, they have reduced the plaques and they have improved the cognitive function and memory. And this comes under the neurotherapeutics. So healthy neurons without plaques with the help of CRISPR-Cas9. So if you are interested that how they have done this, so you can read this paper, this review, in which they have clearly mentioned that how different studies have been performed and how the Alzheimer's disease have been treated with the help of CRISPR-Cas9. Now we'll talk about the SNP editing strategies. So SNP are when we have the single nucleotide polymorphism. So in our genome, we all know that all the human genome are nearly similar. 99% we are identical. We have the identical genome. There is just 1% which is different between different people. So there are some SNPs which are the variants present in the population. So some variants are just normal, whereas other variants can be disease causing. So now how we can edit these SNPs? So there are many diseases which arise from the single base mutations. But keep this thing in mind that not all the variations are harmful, not all the SNPs are harmful, but there are certain SNPs which might lead to the disease. So CRISPR can knock out the defective genes or introduce the protective polymorphisms. Alternatively, these transgenes can replace the defective alleles. So for example, if my gene is faulty, then I can use the transgene, which is the external repaired gene, and I can exchange my already available gene with the transgene. So viral DNA can also be targeted and cut to prevent the infection. So as the bacteria does that, it cuts the double-stranded DNA of virus and prevents the infection. Similarly, we can also do the same. To avoid the viral infection, if we know the viral DNA, then we can simply insert the CRISPR-Cas9 system into the human, and then that system will recognize the viral DNA and it will cleave it. Cas9 will cleave it. So this is how we can use it in the disease states. So now we will see. So SNP editing enables the allele-specific CRISPR-Cas targeting to selectively silence the mutant alleles in autosomal dominant disorders using the nearby SNPs. So in this case, this is the CRISPR compatible PAM, and this is the CRISPR incompatible PAM. So in the PAM, we normally have NGG. So this is the PAM sequence. So Cas9 specifically recognizes this site and cleaves few base pair downstream. But if it is NCG, then in this case, this is incompatible PAM. So in this case, my Cas9 will not be able to cleave it properly. So upstream CRISPR discernible SNP. So in this case, I have the SNP which is upstream of the site. So we can bring about, this is the maternal strand and this is the paternal strand, and this is the downstream intron region, and these are some of the mutations. Now our aim is to treat those mutations, to remove these mutations. Now what we will do, we will do the introduction of Cas9 guide RNA. Guide RNA will enter, it will bind to the site which will be complementary to my uh, the CRISPR RNA complex of the single guide RNA, because we have discussed that how the single guide RNA is constructed. So it has one component which is uh, corresponding to the CRISPR RNA, and another component which is like this twisted edge, which is like a tracer RNA. So both are combined to construct single guide RNA. So this component is complementary to my gene of interest. It will bind it. Now the next thing is that Cas9 can only cleave below the PAM sequence. So it will see whether the PAM is available. So similarly, in this case also the guide RNA has joined it, because what we need is that we want to cut it and we want to remove this mutations. We want to exchange these mutated strand with the correct strand. So similarly, what we will do same in the paternal strand also, and we have this particular uh, CRISPR incompatible PAM in this case. So in the gene editing, what will happen is that we have truncated the maternal strand, and my paternal strand is still normal because it was having the incompatible PAM. So either this incompatible PAM already exists, or we can by ourselves also we can make it incompatible, so that when we use the CRISPR Cas9 system, then Cas9 does not cleave this, because we know that paternal strand is having no mutations. So we don't want to cleave it. We just want to cleave the maternal strand. Now at the same time, we will be using the to repair it, we will be using the homology strand. So in this case, paternal strand can act as the homology strand, and it will also be having the homology arm region, which is complementary to both the sides, because after two double strand breaks, we are removing this segment. So both these arms will be homologous arms. So this fragment, okay, so here they have not properly mentioned, but we will be using the another repair strand with which this segment will be exchanged with the truncation site. So we will repair the maternal strand and we will completely synthesize it. So in that way, the double strand break is repaired, mutations are removed, and here they have only taken the example that in the paternal strand there was no mutations. So this is to show you that this is the control, that we know what is the original sequence. Just for that reason only they have kept it as a paternal strand. But whenever the homology dependent repair is taking place, so we don't mean that whatever is present in between get exchanged and now my paternal strand is left with no intermediate sequence. So here we have just shown that this is acting as a repair strand. So in that way, my maternal strand is now repaired and it will also normally express. Okay, now we'll talk about the pseudogene reactivation. We can also activate the pseudogenes. So pseudogenes are like false genes. So CRISPR-Cas9 mediated reactivation of uricase pseudogene in human cells, it prevents the acute hyperuricemia. So sometimes repairing the defective gene is not possible. For example, I have a gene which is defective, so I cannot repair it. There are some conditions due to which I'm not able to repair it. So in that case, CRISPR can activate the pseudogenes. So pseudogenes are like the genes which are non-functional, and they are like copies, because in our system there are many genes. And for example, if I'm talking about a gene which has undergone duplications, so I have multiple copies of the same gene. But what our genome does is, because in the nature it maintains the balance. So if all those genes get expressed, then there will be the toxicity in our cells. So what it does is it brilliantly make other genes non-functional. It converts those copies into a pseudogene state. That means they are no longer functional, but still they are present. So now what we can do, if we are not able to repair the defective gene, then we will identify that which are the copies of that gene, which are non-functional. So we will treat them, we will make them functional. So these pseudogenes can produce a functional replacement protein, and it is useful when the original protein is toxic due to its misfolding. It expands the strategies for the therapeutic interventions. So in this case, they have, in this particular paper, they have taken the example of the uricase pseudogene, that how we can reactivate that uricase pseudogene so that it can synthesize specific proteins which can help the cell to um, prevent the state of acute hyperuricemia. Okay, so this is how they have mentioned that how now this CRISPR and plasmid and donor plasmid. So this plasmid has entered into the host. Now this particular gene get incorporated into the nucleus, and as soon as it enters into the nucleus, it will get moved to the peroxisomes, because this particular uricase gene might get expressed in the peroxisome. So it will enter there and it will get expressed, the protein will get expressed there, and then it will help the cells to eliminate this condition, to fight this condition. So this is performed in the HEK293 cell lines. So this is not performed in the real host, but these are the experimental studies which were done, that how CRISPR-Cas9 system can be used to reactivate the pseudogenes. Now we'll talk about the gene therapy timelines and different ethics which exist. So CRISPR-Cas, this is a paper in which they have properly talked about how this CRISPR-Cas9 was discovered and what are the ethical considerations, that how we should use it in the genome editing. So early gene therapy trials were done in 1990s and they faced quite setbacks and controversies. Then CRISPR-Cas9 came and it revolutionized the gene therapy possibilities in 2012. So by 2017, human embryos have been edited experimentally, because we have seen that in different disease states, the human embryos could be, which are known as the germline editing, genome editing. So this is the 2017 it was observed, and 2019 CRISPR baby scandal also raised ethical alarms worldwide, due to which that person was also sent to jail. So ethical guidelines remains central to responsible CRISPR use. So these are the ethics of gene editing. So although we have gene editing powers, technologies like CRISPR, it offers us the ability to change the DNA and treat the genetic disease. So this is the basic principle, that if I want to treat the disease, I can use this technique. So there are some promises versus concerns. So while gene editing can transform the medicine, it also raises concerns about the unintended consequences. So for example, if I am changing something in the gene, then I don't know that how it is linked to some other gene, because we know that there are different genes are somehow linked to each other, different proteins are interacting with other proteins, and then they can influence some machinery. So for in one case I am treating it, but we don't know that how in future it can influence the other functions. So still that thing exists. So that's why we need clinical trials and everything to know that whether that gene editing is somehow helpful, or in later term it can influence other genes also. So designer babies, the possibility of using the gene editing for enhancement, it also leads to the ethical dilemmas around the designer babies. Equity and access. So will only the wealthy will have the access to life-changing gene therapies, which deepening the societal inequality. Because we know that gene therapy is not cheap, it is quite expensive. So in that way, it also raises the concern that all the wealthy will be able to access it and the people who are poor will not be able to access it. So in that case also, it raises the conditions. Then what is the ecological impact? So how might altering the organisms might impact the ecosystems and diversity? So in this case, for example, if we are editing some genes, then there are possibilities that it might influence the ecology, the nature, the other organisms. And this gene editing, here I'm only not only talking about the gene editing which is used in the therapeutic purposes. For example, if I want to increase the yield of some plant, as we have seen in the previous sessions. So, for example, soybean plant or rice plant, so we can change certain genes so that its yield could be improved. So in that case, if we keep on editing it, then it might also affect the ecosystem and biodiversity. Maybe those changes might disturb the natural balance of that particular crop. Maybe on later on stages, although I have increased the yield, but maybe later on, the organisms might not be able to digest that particular crop. So in that way, it's important to see that what ecological impacts it could have, and then what is our role. So future biotech leaders must engage in the ethical discussions and responsible research and policy advocacy. So we should must take part in the ethical discussions that what should be legalized, what should not be legalized, and what should be the rules and regulations that one must follow. So in gene editing, we must take the permission before performing it on anyone, any person. So everything need to be keep in mind that we cannot exploit these gene editing methods. We need to keep certain rules and regulations in our mind before proceeding. Yeah. Okay, so now we'll talk about the CRISPR and neurodegenerative diseases, that how the CRISPR has helped in the treatment of neurodegenerative disease. So it is an alternative approach to current therapies which exist. So different neurodegenerative disease such as Alzheimer's, Parkinson's disease, so all of them, primary motor cortex and primary motor neuron disease. So in this motor neuron disease and all these disease, we can, as you can see, this is a normal brain and this is the brain in the Huntington's disease. There are some genes which get mutated. So with the help of CRISPR-Cas9 system, we can repair those genes and we can insert into the viral vectors and then introduce the corrected genetic material into the host. So that is what the gene therapy means. So neurodegenerative disorders such as Alzheimer's, Parkinson's, ALS is autism spectrum disorders, and then Huntington's involve the progressive neuronal death. So in all these neurodegenerative disorders, the neurons get degenerated. That means the neurons number get decreased, they die off. So common causes accumulation of toxic or misfolded proteins. So if folding is not proper, then they will accumulate, they will make plaques. CRISPR can target the disease-causing genes to silence the harmful proteins. So those proteins can be silenced using the CRISPR-Cas9 system. Alternatively, protective variants can be introduced into the neurons. So animals and cellular models show the promising therapeutic benefits. So now, because in animal systems we have seen, so we can use this gene therapy comfortably in the human also to treat these neurodegenerative disorders. So we'll talk about the Parkinson's disease, which is one among the neurodegenerative disorders. So this is the paper in which they have very clearly mentioned that how we can use CRISPR-Cas9 stem cell therapy in Parkinson's disease. So if you're interested, you can go through the detail, but I'll give you the overview. So generally, this Parkinson's disease, it results from the loss of dopaminergic neurons, which releases the dopamine. So in gene editing, targets will be my PRKN, which is the Parkin gene, PINK1, and DJ1. So these genes are responsible for the Parkinson's disease. So we can target these genes. CRISPR can also introduce GDNF, which is the growth factor, or correct the AADC for dopamine synthesis. Strategies include knockout of harmful alleles and knock-in of protective genes. So these are the harmful genes and these are the protective genes. So it can do knockout and knock-in, and it shows the potential to slow the disease progression beyond the symptomatic treatments. Okay, so in this case, everything is mentioned that how different genes which are involved in the mitochondrial function, so how they are altered, how it leads to the disturbance in the mitochondrial population in the cell, and how the reactive oxygen species leads to mitochondrial damage, and how different other genes are formed, different the proteins are synthesized, which ultimately leads to the neurodegeneration. So for and similarly, example is given for alpha-synuclein aggregates, which the aggregates are synthesized in the cells, so which leads to the impaired proteostasis in which the balance of the proteins are disturbed. Similarly, here also they have shown that different other proteins are synthesized, which leads to the uh, means which leads to the disturbance in the balance and ultimately leads to the Parkinson's disease. So all these genes which are leading to this particular state, they can be cured with the help of CRISPR-Cas9 method. And now comes the delivery approaches, that how to deliver the CRISPR therapies into the host. So first one is ex vivo delivery. That means patient cells are edited outside the body and then reinfused. Second thing is in vivo delivery. That means CRISPR introduced directly into the tissues. And for this delivery, we use the viral vectors such as AAV, adeno-associated virus, adenovirus, or lentivirus, and these are commonly used for high efficiency. Then comes non-viral methods such as lipid nanoparticles, RNP delivery. So this is the lipid nanoparticles and the gold nanoparticles, which reduce the immune risk, because in the case of viruses, there is still a risk that they can lead to some other immunogenic behavior of the organism. So in that case, non-viral delivery system is a little bit better in case of immune risk, although it's less efficient than viral vectors. And delivery method must balance the efficiency, specificity, and safety. And these are some of the physical methods. So whenever we are doing the delivery, we should keep these things in mind, that its efficiency should be better. Although its efficiency is better, but it should also be safe and it should be specific. That the region where we want to have the influence of that system, there only target the specific target gene. It should not bind to off-target site. So that comes under specificity. So all these things need to be kept in mind while we are using these delivery systems to introduce the CRISPR-Cas system into the host. Now we have shown the difference between ex vivo and in vivo workflows. So ex vivo, extract the patient cells, culture them, CRISPR edit, expand and reintroduce. So this is the ex vivo system where we are changing things externally and then inserting the correct thing into the cell. It is safer as edited cells can be screened before the patient's use. If we talk about in vivo, then in this case, we deliver the CRISPR system locally to the tissues. This is faster, but it carries the risk of off-target edits inside the body. And choice depends on the disease type and target tissue accessibility, that how easily we can access that particular target tissue. So here is an example of in vivo, and here is an example of ex vivo workflow. So in vivo, we use these vectors and we directly insert the CRISPR-Cas9 into the host, and then within the host, it will treat that gene. Whereas in this case, we extract that HSPs, human HSPs, and then T cells, and we collect them. We transduce the cells with the transgene, we correct that particular gene, and then we introduce the modified cells back into the patient. So in this system, we already know that we have treated it. But in the in vivo system, there are chances that off-target site binding might take place. So we are not sure with it. So these are some of the trade-off between both these approaches. So it depends that whether what is the condition, and we decides that whether we should choose in vivo or ex vivo. Now there is another study of the Duchenne muscular dystrophy. So this is a paper in which they have mentioned that how the Duchenne muscular dystrophy can be treated with the help of CRISPR-Cas9 by specifically deleting the exon 44 mutations in mice and human cells. So this Duchenne muscular dystrophy, it is a lethal X-linked disease which is caused by the mutations in dystrophin gene. So this is the gene. Lack of dystrophin leads to progressive muscle wasting and weakness. So if this protein is not properly formed, then it will lead to the weakening of the muscles, and it will ultimately lead to the Duchenne muscular dystrophy. So CRISPR corrects this Duchenne muscular dystrophy by reframing or skipping the exons in the genes. Skipping exon 43 and 45 restore the reading frame. Patient-derived immunopluripotent stem cells shows the restored dystrophin after editing. CRISPR-Cas9 correction of exon 44 deletion in patient cells and mouse models highlight a promising step towards the clinical application. So in this example, I'll tell you that there are multiple exons which are present in this gene, which is the dystrophin gene. So now if exon 44 is present, for example, that in that case, targeting exon 43 in 3.8% patient coverage. So now in this case, exon 43, 44, 45, 46 is present. So if we skip exon 43, then exon 44 is Okay, so in this case, they have already exon 44. So in this case, they have already deleted exon 44. We don't have exon 44. So if exon 44 is deleted, then in this case, 43 and 45 will combine. So because they will combine, so because there is in between deletion, so when their sequence combines, there will be a generation of the stop signal. Because let's assume here we have the, uh, for example, UAA comes when we joins them together. Stop codons come due to the sequence frame shift, due to change in the frame of the sequence, the stop codon comes. So in this state, if exon 43 and 45 combines, then it leads to the stop signal, and it will ultimately lead to the uh, premature termination of this gene, due to which my protein is not properly synthesized. So there are two ways. Either I can target my exon 43 or I can target my exon 45. So if I can target both of these exons, then in both these cases, we can correct the deletions, or we can correct the frame which has changed. So these are two different ways. We can reframe the exon 43, or we can skip the exon 43, or we can skip exon 45, or we can reframe exon 45. Means we are doing this in such a way that 43 and 45 do not combine together in their original forms. So either we can delete one of them, or we can reframe either one of them, so that the stop codon is not observed. So not just 43 and 45. In many papers, you might see some other exons also. So main thing what we are doing with CRISPR-Cas9 is that we are deleting those exons, which leads to premature termination of this gene. So if there is loss of any middle exon, which is leading to the short or terminated protein, then we can treat that condition, that why the terminated product is synthesized. Either there is generation of stop codon, and which exons are leading to the premature termination. So we can delete those exons, or we can change those exons with the CRISPR-Cas9 system. Yeah, so this was another paper in which they have talked about the exon 52 deletion. So in this case, we talked about 43 and 45 case. Similarly, in this case, exon 52. So in this case, if exon 52 is present, then the dystrophin is synthesized. But if 52 is missed, then 51 and 53 combine in such a way that it leads to premature termination. So dystrophin gene do not function properly. So in this case also, they have mentioned that how we can treat it. So this is the paper. If you're interested, you can go through this. But the main idea which you should take from here is that with the help of CRISPR-Cas9, we can specifically target those exons. We can either remove them, or we can make certain changes in them so that it could be reframed and proper dystrophin protein can be synthesized. Now designing the single guide RNAs for the DMD correction. So this talks about that how we can design the single guide RNAs so that we can correct this. So this is the mechanism. So let's assume single guide RNAs that target the splice donors and acceptor sites flanking the defective exons. PAM sites determine where the Cas9 can cut. So multiple single guide RNAs tested to identify most efficient candidates. Precision design ensures the correct skipping and minimal off-target edits. Effective design is critical for therapeutic success. So this is my intron and this is my exon. So I'm doing it at the level of DNA, because we know that there are these type 2 CRISPR-Cas systems, they target the DNA sequence. So at the level of DNA, I don't have the just the exons. We also have the intronic region which is present. So similarly, in this case, this is the intronic site and this is the exonic site. So they have mentioned that this single guide RNAs, they target the splice donor site. So this is the splice donor and acceptor sites. Yeah, so in the intron, we have the splice donor and acceptor sites from where the splicing takes place. So this is the 5' site and this is the 3' site in this case. Similarly, in this case also. So it targets those sites. The PAM is marked in red. So this is the PAM sequence. And this is my exon 45. So the PAM region is present here. So this is CTT and this is GAA. So now we want cleavage here such that this exon get removed. So similarly, in the case of single guide RNAs, they are designed in such a way that they are complementary to the splice donor and acceptor sites. They will target it and they will remove these sites so that this particular exon gets skipped such that spliceosome can split it. It does not get into the mature product. Now the validation of DMD gene editing. So edited immunopluripotent stem cells are differentiated into the muscle cells. Western blot shows that the expression of dystrophin is restored because after doing the knockout of this particular exon, we will see that how it influenced the dystrophin protein. Because previously, when this protein was getting synthesized, and due to the stop codon, because we have multiple exons after this also, so dystrophin protein is quite big. But if it gets terminated here only, then the protein is short. That means it is non-functional. It will not properly fold and perform function. So by either removing both either of these exon or by reframing them, the proper protein is getting synthesized. So how to test it? So that's why they are doing the Western blotting, in which they are checking for the that where the bands are obtained. What is the length of the protein? Either it is short or it is long. So this is for the control. They have compared it with where the band is observed. So Western blot has shown that it has restored the dystrophin protein. So immunostaining also confirms the correct localization of protein in the cell, and this demonstrates the functional correction, not just the DNA repair. So functionally also it's corrected because protein is now getting synthesized. So this is the proof of concept for the translating CRISPR into the patient therapies. And this is how the staining experiments have also shown that how the dystrophin gene, this is the wild type state, and this is the state when 45, 43, in this case 44 is deleted. So 43, 45 when combines, it leads to the stop codon due to which the proteins are not properly expressed. So as you can see, this red dots are not evident. That means the dystrophin gene is not properly expressed. Whereas now they have deleted, they have reframed exon 43 here and deleted exon 43 here. So in this case, again the expression is restored. So dystrophin expression is increased. Similarly, here also they have either removed 45 or they have reframed it in such a way that dystrophin protein is properly synthesized. So these are the ways how we can validate that my genome editing has worked properly, whether it is uh, working at the target site or it is working at the off target site. So this result shows that yes, it is correctly working. It has correctly made the deletions at the correct sites. CRISPR gene drives. So we'll talk about what are the CRISPR gene drives. So gene drive means spread the genetic changes faster than the normal inheritance. Cas9 cuts the wild type allele in the heterozygotes. Homology-directed repair copies the drive alleles onto both the chromosomes, and nearly all the offsprings inherit the modified allele and potential to alter the entire population of an organism. This is referred to as gene drive mechanisms. So as we say in inheritance, we inherit something. So in inheritance, in this case, as you can see, the altered gene does not spread well. This is the altered gene and this is the normal gene. The altered gene is not properly spreading. But in the case of gene drive inheritance, as you can see, altered gene is always getting inherited at each level. So this is how, with the help of gene drive, we can lead to the uh, faster genetic changes. So this is the gene drive mechanism. This is the target sequence. Here we have the Cas9 single guide RNA and the cargo. So cargo is the one which we want to insert. So now what will happen is that my Cas9 will come and my single guide RNA is complementary to my target DNA. So it will bind to that particular target site. It will bring Cas9 and it will search for the and it will repair that particular site. So now what my aim is that I want to insert these elements into my target site. So with the help of this Cas9 complex, we will cut it here. So it will identify the PAM site and make double-stranded break. Now we have this region. So these dark lines, they are complementary in both the sides, so they will act like the homology arm. So this particular region will be copied here. So it will repair it, and both of these chromosomes, they are passed to the next generation with the gene drive cassette. So this is my gene drive cassette. Similarly, here also it is shown, this is the Cas9 guide RNA and the cargo. And this is the cut site where the cut will take place, because cut will only take place at the target site, which is complementary to the guide RNA. So it will repair it. This is the allele 1 and this is the allele 2. So after the cut, because both of them has the homology arms, so this component get transferred to this particular allele also. So this is how the gene drives work. Now, what are the applications of these gene drives? So, eradicating malaria by targeting mosquito populations. Second is controlling the invasive species that damage the ecosystems, spreading the disease resistance in crops or livestock, and then restoring the sensitivity to pesticide or antibiotics. And major ecological and ethical debates remain unresolved. So, we can control the invasive populations and we can protect the endangered populations using these gene drives. So there could be eradication drives where we are eradicating something, such as we are eradicating mosquitoes, we are eradicating pest diseases, pest species. Suppression drives means we are, for example, if something is spreading widely. So spreading mildly deleterious mutations in invasive populations, so we can suppress them, which leads to the reducing the height of invasive common ragweed to decrease its competitive pressure on the native plants. So these are some of the examples. If you're interested, you can go through them in detail. I have given the link of the paper from where I've obtained this figure. So these are the applications of the gene drives. They can be rescue drives where we can spread the beneficial mutations in the endangered populations. So in this case, the spread is quite fast as compared to the normal inheritance. So in that way, they can act as the rescue drives. Now, other than genome editing, we can use CRISPR for other mechanisms also. So, catalytically dead Cas9, which is known as dCas9, it binds the DNA without cutting it. It is used for the gene regulation, in which CRISPR-I, which is involved in the repression or inhibition. Then we have CRISPR-A, which is activation. So, dCas9 it is fused to the epigenetic modifiers, which will alter the histone marks or DNA methylation. CRISPR-based imaging tracks chromatin organization in the live cells, and it expands the CRISPR toolkit into the gene expression and epigenome research. So this is the CRISPR-Cas nuclease. We've already seen that how there is a single guide RNA, and this is another example of Cas12a in which we have the CRISPR RNA and a tracer RNA. So it makes the double strand cut in the genomic DNA or in the double-stranded DNA. So this is how double-stranded DNA is present. It can now repaired by NHEG or HDR, and depending on the repair mechanisms, the output can be different. Now it can also work as base editors, in which we can edit certain bases, and so we can change the bases from A to G, C to T, A to C, or C to G. So this is how the editing works. So in this case, we have added the deaminase, and this is the editing window. So with the help of deaminase, because we mentioned that in this we use the dCas9, because normally if Cas9 is present, it makes a double strand break. But now we don't want the double strand break to take place, so we are using dCas9, which is the dead Cas9, which will not break, and we fuse it to epigenetic modifiers. So in this case, epigenetic modifier is deaminase. So deaminase will deaminate it, which will convert the A to guanine. So this is the editing window. This is how the base editing takes place. Second thing is prime editors. So we can also bring about the 40 base pairs insertions or 80 base pair deletions, point mutations within 30 base pair of Cas9 mixed site. So all these changes can be brought with the help of certain modulators which are responsible to bring about these mutations. Similarly, we can use it for the transcriptional modulators, in which we can either use VP64, which is for the activation, and CRAB, which is for the CRISPR inhibition, or you can say repression of the genes. Then it can also act as RNA editors, in which we can attach the adenosine deaminase to it, and it will edit the mRNA. So it can edit the adenine to the inositol or inosine. which is the conversion. So in this way, I am editing the bases at the level of RNA. Okay, now we'll quickly go through the base editing concept and its applications. So base editors use the Cas9 with the deaminases. So what deaminases do? Deaminases remove the amino group with which we can convert the base from one form to another. So cytosine can be converted to thymine and guanine can be converted to adenine on the opposite strand. So this is the example. These are the cytidine deaminases. So as cytidine deaminase, that means they will remove the amino group from the cytosine or cytidine. Cas proteins are involved because they are taking these particular components along with the this guide RNA into the that site, target site. These are adenosine deaminases. So what cytidine deaminase is doing? It is converting C to U. Now U is there, but whenever the another strand will get synthesized, then it will have the T. So that's why we say C to T transition. But actually C is converting to U and in its complementary strand we will have T. That's why C to T. Similarly, in the case of adenine base editors, A will get converted to I, and now my inosine will bind to guanine. So in that way, A is converting to G. So in my normal strand where A was present and its complementary strand should have T, but now because A has changed to I, so my complementary strand will have G. So that's why it is known as A to G conversion. So its application are SNV correction. So we can also correct SNVs because, for example, if A is the product of mutation, so I can remove this mutation by using these base editors. So in that way, I can correct the single nucleotide variations. We can do the gene regulations, and in alternative splicing also it helps. Yeah, so here is how it has shown that how the cytidine deaminases converts C to U, and then in the next round, when the product is synthesized, so in place of U, we will get T. Now we'll talk about the current status and challenges in the DNA-based editing tools. So although these DNA-based editors can be applied as versatile tools for therapy and disease modeling, and these CBEs convert specific, so these base editors can convert specific codons like CAA, CAG, CGA, CGG into the premature stop codons because we know that cytidine deaminases, they convert cytosine into the uracil. So if it is changed with U, then it will become UAA, UAG, and UGA and UGG. So these are like stop codons. So in that way, if stop codon is present, then it will lead to the knockout of gene function because it will terminate the protein synthesis. It will not allow my protein to synthesize properly. It will stop the termination. So it will terminate my protein. So that's why we can knock out the genes using these editors. Then if we talk about adenosine-based editors, then they convert ATG into the known start codon. So instead of ATG, now we have GTG. So transcript means the synthesis will not start because these codons are responsible for the protein synthesis. So start codon is not recognized, so protein synthesis will not start. So in that way, it can lead to the silencing. Similarly, it can also convert premature stop codons into the amino acid codons. So for example, previously I have TAA, so I can change this A and I can convert it into something else. So they have changed it to CAA. So now CAA is not stop codon, so protein will continue to synthesize. So we can restore the protein synthesis, which was previously stopped here. Then we can use CVEs to edit the splice acceptor sites by converting G to A by altering the splicing. So if G is changed to A, then it will also lead to change. So generally, A here will lead to the skipping of this particular exon. In this case, where G was present, so this exon was retained, but now the splice site, which was recognized by spliceosome, now it is not present, so spliceosome will not recognize it, and it will skip this exon. So this is how the base editors can be used to skip these sites, and these base editors are attached to the CRISPR systems with the help of CRISPR-Cas9 system. It is now bringing out this effect. So these are some of the applications and challenges of the DNA-based editing. And if you want to read it in detail, then you can go through this review. Then comes prime editing. So in the base editing, we were only talking about one single base. In prime editing, we combine the Cas9 nickase with the reverse transcriptase and a PEG RNA. So this PEG RNA guides Cas9 and it encodes the desired edit. So it can introduce all the 12 base substitutions plus insertions and deletions. So in this case, we are either bringing about larger component insertions or deletions into the segment. It is more versatile than base editing because in this case there are only few off targets than the HDR, and it expands the scope of precise genetic modifications. So this is the example that how the Cas9 nicases cut one strand of the DNA and then reverse transcription of the RNA incorporates the desired sequence into the target DNA. Edited strand is incorporated, original DNA is cleaved by cellular endonucleases, and unedited strand is repaired to match the newly edited sequence. So both the strands contain the desired edit. So this is the edit which we wanted to insert. So this is how the prime editing works, that in the given double-stranded DNA, we can insert of our something region of our insert, and from where this region was obtained. So this was obtained from um, this particular RNA which we had, single guide RNA. So it was also having some cargo. So this cargo sequence which we wanted to insert into the DNA. So this cargo sequence got incorporated now. So this is how we can repair, we can add the newer fragment. There was reverse transcription. In reverse transcription, the RNA get converted to DNA. So my desired sequence, which was the cargo sequence attached to my guide RNA, so it is converting to the DNA and it is getting incorporated here in both the sites. Now, if we talk about the CRISPR's role in the transcriptional regulation, then we have discussed that if uh, CRISPR-A means activation and CRISPR-I means it is doing inhibition. So if VP64 is fused with the DCAS9, so DCAS9 is dead Cas9 which do not cleave. So in this case, if VP64 or P65 is present, then it leads to activation of the gene. So in activation, the transcriptional status will be green. That means transcription rate is high. More and more transcription is happening. Whereas if we use the CRAB domain along with DCAS, then it leads to the CRISPR interference or inhibition, which leads to the suppression of the transcriptional process. So it has as low as possible. So in this case, the gene is not getting transcribed. No RNA is getting synthesized. So it is leading to inhibition. It is leading to stimulation. Okay, similar thing is mentioned here, activator and inhibitor. So in activator, transcription will take place. In inhibitor, the transcription will not take place. Now, what are the future prospects of CRISPR technology? So computational tools and AI is improving the single guide RNA design and off-target predictions. There are new Cas variants which are coming up, such as Cas12, Cas13, and Cas14, which expand the DNA RNA editing capabilities. Because when we were talking about Cas9, then it can only target the DNAs. So now these variants can target both the DNA and RNA. RNA targeting CRISPR offers the therapeutic options for the viral infections because there are certain viruses which are RNA viruses, so we can target with the help of these newer Cas proteins. Agricultural applications include climate resilient crops and disease resistant livestock. Then comes ethical, regulatory, and ecological concerns, which remain central for human and environmental safety. So these are the applications which I have obtained from this paper. So if you're interested, you can go through this link and you can read in detail that what are the applications of CRISPR technology. And this is the entire summary of today's session. So this is a review which is very nice, in which they have very clearly mentioned detailed past, present, and future of CRISPR genome editing technologies, starting from the that how we can use it, different things, different effects, different editing which we can bring about with the help of CRISPR technology. And along with this, they have also talked about that how we can do gene knockouts, we can bring mutations, how we can use it in screenings, and how we can do ex vivo and in vivo gene editing, which we have already discussed. So this is the basic research and this is the therapeutic purposes of this particular CRISPR-Cas9 technology. And this is the functionality of all the means what kind of editing can be done with the help of CRISPR-Cas9 system. So if you're interested, you can go through this. We have already discussed it, so I'll not go into this again. So now we'll practice the MCQs. So sorry. Okay, so now we'll practice the MCQs. So first question is, what purpose does the CRISPR-Cas system serve in the organisms where it is naturally present? Option A, defense against viral infections. Option B, regulation of gene expression. Option C, repair of the damaged DNA. And option D is synthesis of essential proteins. So in this question, they are asking that what purpose it naturally serves. So the answer is defense against the viral infections, because we have already seen that in the E. coli study, when the scientists were performing study on the E. coli or the different prokaryotic organisms, then they observed that there are certain sequences which are present between the repeats, which are around 35 nucleotide in length, which are known as the spacer sequences, and they are similar to the viral DNA. So in that way, this particular viral DNA sequence is acting like a memory of the previous infection. So now whenever such virus again attacks the host, so these spacer elements, these are known as precursor CRISPR RNA. So they get activated, they get converted to the CRISPR RNAs, and then they combine along with Cas9 protein. They identify these sites in the target sequence. They identify that particular target sequence, and the Cas9 will do the cleavage. It brings about double strand break of the DNA element. So in that way, they protect themselves from the infection of the virus. So the right answer is defense against viral infections. Now next question is, what is the primary function of tracer RNA in CRISPR-Cas9 system? Option A, to degrade the viral RNA. Option B, to guide the Cas9 proteins to the target DNA. Option C, to repair the damaged DNA. And option D is to synthesize the new proteins. So the right answer is to guide the Cas9 protein to the target DNA. So without the tracer RNA, this Cas9 is inactive. It cannot do the RNA cleavage. But with the tracer system, it can, along with the this CRISPR RNA binding and DNA binding, so both of them makes the RNA duplexed, and they helps the Cas9 to identify this target site and make the cleavage. So that's why it guides the Cas9 protein to the target DNA site. Next question is, which of the following viral vectors is widely used in CRISPR gene therapy because of its high efficiency of delivery? Option A is lentivirus, option B is retrovirus, option C is herpes simplex virus, and option D is adeno-associated virus. So this also we have already discussed that among all the viruses, adeno-associated viruses are the ones which are widely used because of they are safe, they have low immunogenicity, and they have high delivery efficiency. So among viral and non-viral vectors, viral vectors have higher efficiency, and among all the viral vectors, adeno-associated virus is the one which has better, which is better among all of them. Whereas if we talk about lentivirus, it is efficient for integrating genes into the dividing and non-dividing cells, but it is less preferred in CRISPR. If we talk about retrovirus, the viruses which has RNA as genetic material, so they have high risk of insertional mutagenesis. And if we talk about herpes simplex virus, so although it has a large capacity, but it is less commonly used. The right answer is adeno-associated virus. Now next question is, what is the full form of CRAGE in the context of genome editing? Option A, chassis responsive active genome editing. Option B, cellular recombination and genetic engineering. Option C, chromosome related active gene editing. And option D is chassis independent recombinase assisted genome engineering. So the right answer is option D, which is chassis independent recombinase assisted genome engineering. So what chassis means? It simply means the species. So it is independent of species. In this, we use the recombinase assisted genome engineering. So in this, we have the LOXP sites. So it is based on the recombinase work, that there is a chromosome, and this is the donor, this is the recipient. So as we send this particular element into the recipient, then with the help of these transposases and these LOXP sites, this particular element get into the chromosomal DNA. So as you can see here, that this particular region get integrated into the chromosome. And we can also insert the Other elements such as BGC integration can also be done. So LOXP sites are present. So we are using the recombinases. With the help of recombinases, we are inserting the components of interest into the chromosome, and this is irrespective of the species or the chassis. So that's why it is known as chassis independent. It is a synthetic biology platform which enables the rapid and efficient integration of large DNA constructs into diverse microbial hosts. So if we talk about traditional methods, then they are limited only to the model organisms. But in the case of CRAGE, it works across multiple chassis species by using recombinases to insert the genetic circuits at the defined genomic sites. And this system accelerates the strain engineering, allowing the researchers to explore the natural product biosynthesis and metabolic engineering and functional genomics in known model microbes, the microbes which are not well studied. So for that particular reason, we can use them. Next question is, what causes Duchenne muscular dystrophy? Option A, overexpression of the myostatin gene. Option B, gain of function mutation in the dystrophin gene. Option C, loss of function mutations in the dystrophin gene. And option D is loss of function mutations in the myostatin gene. So we have currently, means in the recent topic only we have talked about this. So the right answer is option C, which is loss of function mutation in the dystrophin gene. So we already know that due to certain exons, the premature termination takes place, due to which this gene is incomplete. So due to incomplete genes, muscles are weak. So this leads to the DMD disease. So that's why the right answer is loss of function mutations. Next question is, which of the following enzyme is fused with the DCAS9 for the construction of cytosine-based editors? So option A is DNA methyltransferase, option B cytidine deaminase, option C adenosine deaminase, and option D is reverse transcriptase. In this case also, it is quite straightforward that cytidine deaminases, because with the help of deaminases, they convert the cytosine into the uracil, and now this uracil, whenever the complementary strand will get synthesized, so this uracil will bind to thiamine, so it leads to C to T modifications. So this is the cytosine base editor which is using the cytidine deaminase, and bcas9 means the cas9 is dead. That means there will be no cleavage of the double stranded DNA and just the mutation will only be observed. Next question is, the tripartite activator domain VPR does not contain which of the following elements? Option A, RAX from the zebrafish. Option B, VP16 from herpes simplex virus. Option C, RTA from Epstein-Barr virus, and option D is mammalian P65. So you need to tell that this VPR sequence, it does not contain which element. So as the name suggests, VPR. So from here only you can get some hint. So let's see the right answer. So the right answer is RAX from zebrafish. So this particular element is not present in this domain. So this VPR domain, it is having this is a tripartite transcriptional activator domain which contains, which is engineered to boost the gene expression. So it is formed by fusing the three strong activation domains. First one is VP64, which is derived from the herpes simplex virus. So VP64 is derived from the VP16 herpes simplex virus. So VP16 is the protein. So from that protein, this is the domain. Then comes P65, which is a mammalian NF kappa B subunit. And then there is RTA, which is from the Epstein-Barr virus. So from Epstein-Barr virus, so these are the tripartite activator domains which synthesize VPR domain. And when it is attached to the deactivated Cas9 protein, which is DCAS9 or the other DNA binding proteins, it can powerfully activate the transcription of target genes without cutting the DNA. So it is used in the CRISPR-A, which is CRISPR activation. So till now we were only talking about VP64, but this VPR composed of these tripartite domains, so they together act as a strong activator. Okay, so next question is, what are the two catalytic domains of CRISPR-Cas9? This also we have discussed in the beginning of this session. So A is PAM and RUB9. Option B is HNH and RUB9. Option C is HNH and RUBC. And option D is PAM and RUBC. Okay, so the right answer is option C, which is HNH and RUBC. These are the two domains, and these are the two nuclease domains. So in which HNH cleaves the DNA strand complementary to the guide RNA, whereas roof C cleaves the non-complementary strand. So this is the roof C and this is the HNH. So this is my strand which is complementary to my guide RNA, which is cleaved by HNH. This is the non-complementary strand and this is cleaved by roof C. Okay, so the next question is, what role does the PAM play in CRISPR-Cas systems? Option A, influences the efficiency of DNA cleavage. Option B, involved in RNA synthesis. Option C, crucial for the acquisition and interference. And option D is not essential for the CRISPR-Cas system functions. Okay, now let's see the answer. So the right answer is option C. That means this PAM is crucial for the acquisition and interference. So the first option was that it influenced the efficiency of DNA cleavage. So it has nothing to do with the efficiency because it is more than just about efficiency. It is required for the recognition because below it the Cas9 cleaves and then involved in RNA synthesis. So this is completely wrong. And then PAM sequences are essential for the Cas9 proteins to distinguish target DNA from the self DNA and enable both spacer acquisition and target cleavage. So this is the right option. And this is the PAM distal and PAM proximal motif, and this is the PAM site. So the regions which are near to PAM, they are known as, this is known as seed region, and this is the tail region, and this is the PAM proximal region, and this is the PAM distal region. So next question is, what is the role of inteins in the protein processing? Option A, protein degradation. Option B, protein splicing. Option C, inhibition of gene expression. And option D is enhances the protein synthesis. So in this case, the right answer is option B, that is protein splicing. Now many of you will be confused that what are intron and what are inteins. So in the DNA, we have introns, exons. So similarly, the inteen term is used at the level of proteins. So whenever in the RNA, after the premature mRNA enters into the cytoplasm, so before it, there should be processing of this RNA in which the premature mRNA converts to mature RNA. So in that case, introns are removed, which process is known as splicing. So when splicing takes place, introns are removed and then exons are joined and mRNA is now ready for translation. But at the same time, there are some components which are not removed in splicing, which forms the protein, and then this protein is spliced. So the component which is removed after the splicing of the protein is known as intine. So teen words here means at the level of protein. And intine means that the elements which are removed at the level of proteins. So that is the main difference between inteins and introns. Introns are removed at the level of RNA splicing, whereas inteins are removed at the level of protein splicing. Okay, so this is the another key difference which I've mentioned. Okay, so now I have kept 20 questions. So I'll quickly go through them so that I don't waste much of the time. So 11th question was, what was one of the Alexander Bolotein's key finding about CRISPR in 2005? Option A is that CRISPR exists only in the yeast genomes. Option B was certain CRISPR sequences matched the fragment of bacteriophage DNA. Option C is CRISPR elements were proven useless in bacterial defense. And option D is PAM sequences were reported to be longer than 15 nucleotides. So the right answer is that they match the fragment of bacteriophage DNA. This we have discussed that with his study only, we have identified that what is the role of those spacer elements which are present in between the repeat sequences. So when he matched them, then he observed that these are the bacteriophage DNA. And then later on, it was identified that it was acting as a adaptive immunity for the bacteria. Okay, next question is, in 2007, what did Berengo and colleagues demonstrate about CRISPR-Cas system? Option A, CRISPR regions were present exclusively in archaeal genomes. Option B, CRISPR added together with Cas proteins provided immunity to bacteria against viral infections. Option C, viral sequences inserted into CRISPR had no functional role. And option D, CRISPR systems were unable to adapt against new bacteriophages. Yeah, so this is also in continuation with the previous study. So in this, they have identified that these Cas9 proteins, they provide the immunity to bacteria against the viral infection, because now we know that CRISPR and Cas works together. So this is the role of the spacer elements, that it is providing the immunity to the bacteria. Next question is, which of the following best describes the type 2 CRISPR-Cas9 system? So it acts on both RNA and DNA equally. It is characterized by Cas9, a large endonuclease enzyme. It belongs to class 1 CRISPR systems, and it completely lacks Cas1 and Cas2 genes. So you need to select one which best describes it. So the right answer is, it is characterized by Cas9, which is a large endonuclease enzyme, because we discussed that type 2 falls under the class 2 system, which uses only the single effector Cas protein, and specifically type 2 involves the Cas9. Next question is, what is the function of CRISPR locus in bacteria? Option A, to break down viral proteins directly. Option B, to record the viral DNA snippets for future RNA guided defense. Option C, to block the expression of Cas proteins. And option D is to attack the viral DNA sequences. Sorry, bacterial DNA sequences. Okay, so it's also straightforward. The right answer is option B, to record the viral DNA snippets, to incorporate into the repeats, which were the uh, the palindromic repeats, and these will act as the uh, okay. So these viral DNA snippets, they act as the spacer elements in between those repeats, and later on it will act as the RNA. It will help in the RNA guided defense, because whenever the same virus will attack, then it will act as the guide RNA, and it will protect the bacteria from the infection. Next question is, in engineered CRISPR-Cas9 gene editing, what role does the guide RNA play? Option A, it cut the DNA strand itself. Option B, it directs the Cas9 to the exact DNA sequence to be modified. Option C, it substitutes for the Cas9 protein. And option D, it blocks the Cas9 from attaching to DNA. Yeah, so this is also quite straightforward, that the guide RNA, it directs Cas9 to the exact DNA sequence to be modified. And how it does it? Because this guide RNA is complementary to the target DNA site. So it will bind to the target DNA. So now Cas9 knows that where to target, where to make the cut. So it will also look for the PAM site. But the main thing is that it will guide the Cas9. Next question is, what is the likely result of NHEJ repair after Cas9 generates a double strand break in a coding region? Option A, the edit is always precise and error free. Option B, indels may arise, leading to frame shifts or premature stop codons that knock out the gene. Option C, it guarantees homology directed repair will occur. Option D, the DNA sequence remains completely unchanged. Yeah, so this is also straightforward, that it is never precise. So option A is completely wrong. Option C, that it guarantees, so this is not true. Then it says DNA sequence remains completely unchanged. This is also not true, because it leads to indels, frame shifts, premature stop codons. It can lead to knockout of the gene. Next question is, how can CRISPR-Cas9 produce the gene knockout? Option A, by introducing mutations to NHEJ alone. Option B, by breaking the gene and allowing the random indels. Option C, by inducing a double strand break and repairing it by HPR with the donor DNA. Option D, by inserting the LOXP sequence for recombination. So here we need to tell about how we can use it to gene knock in. We want to insert the gene. So this is also straightforward, that whenever we want to insert the gene, we generally repair it with the homology directed repair, in which we have the donor DNA and its homology arms, so through which we can knock in the mutation of interest, or we can insert the gene of interest. So we can do the knock in. We are inserting something. Next question is, what is the main role of catalytically inactive Cas9, which is dCas9, when fused with the other molecules? Option A, to directly cut the DNA. Option B, to control the gene expression at specific sites. Option C, to strengthen the RNA interference activity. And option D is to replicate the chromosomal DNA. So it is also straightforward, to control the gene expression at specific target sites, because dCas9 does not cut. And then RNA interference activity, so this is also incorrect, because RNA interference pathways are quite different. And to replicate, this is also completely wrong. So right answer is option B. And in this we have seen the example of CRISPR-A and CRISPR-I systems, where activation and inhibition, both systems we have seen. Next question is which option correctly reflects strategies for reducing the CRISPR-Cas9 off-target activity? Option A, alignment tools like BWA or bowtie predict off targets perfectly with CRISPR like mismatch tools. Option B, off target binding is more likely if followed by PAM such as NGG or NAG. Option C, limiting mismatches to 3 ensures no off target occurs. And option D, Cas9 cannot recognize any alternative PAM sequences. So the right answer is option B, which says that off-target binding is more likely if followed by PAM such as NGG and NAG, because the canonical um, PAM is NGG. So there are some alternatives such as NAG which increase the chance of off-targets. Okay, so the last question is how do CRISPR-based gene drives typically function? So we have already talked about gene drives. So option A is high threshold drives spread rapidly with very few individuals. Option B, low threshold drives require more individuals than the natural population to succeed. Option C, they bias inheritance by converting heterozygotes to homozygotes through double-strand break repair by homologous recombination. And option D, drive alleles are rarely passed to the gametes, limiting the spread. Okay, so the right answer is option C, because we know that in comparison to the normal process of inheritance, gene drives very quickly spread. So they bias the inheritance, and how it does it, they convert the heterozygotes into the homozygotes. So that is the main reason behind this CRISPR-based drives. They cut the wild type allele and the cell repairs it using the drive allele as a template, so ensuring the biased inheritance. Okay, so that's it for today's session. So in the next session, we'll be talking about the VK topics. So if you have any doubts, then you can comment down below this video, or maybe you can write your doubts in the discussion forum where I'll be addressing your doubts. So for now, I'll end this session. Thank you very much.